No, I'm not sure if it's a meeting program. We do it.
I want to welcome everybody to tonight's Ocean Science Lecture, which will be given by Dr. Joshua Voss. Most of you know me, I'm Dennis Hansack. I coordinate the series. And I'm also happy, if you don't know me, to hear your comments and tell me what you like about the talks, or what you'd like to see in the future. Now next week, there will not be a lecture. Okay, we're taking a break. It's actually a planned timeout. Yes, the Olympics. We have all the main events next week. I can check the schedule. Bobsled, figure skating, and my favorite, curling. The curling finals are next week. So we learned 4 years ago, do not compete with curling. Now, the other reason we're doing this is to give our next speaker, who's in the back row, Dr. Tammy Frank, an extra week to prepare for talk. Actually, that's not true. She's away at another meeting next week. She's going to speak on ocean exploration and deep sea research, trials, tribulations, and discovery. Many of you know Tammy from previous talks. You know she's a wonderful speaker. She's going to talk about the deep sea, which is the largest habitat on Earth, with many opportunities for discovery and exploration. She's going to talk about her recent discoveries on her research expedition and share the challenges and excitement of research in the deep sea. Now, I've learned many things from Tammy's talk. One of the things that I like the best is the disgusting hagfish. It's one word, right, Tammy? And so that will be featured in next week's talk, I'm sure. I also want to mention that tomorrow night there's a lecture down at the Pelican Yacht Club. It's part of the Smithsonian Marine Science Lecture Series. It's the Christian Sampler, who's the head of the Natural History Museum. He's here in Fort Pierce. He's going to give a talk. And some of the highlights will be, for example, the ocean exhibits, the Tree of Life project, and a few others. And we have some flyers out in the gallery on that. Now it's a great pleasure to introduce tonight's speaker, Joshua Voss. Josh grew up in Florida, near Orlando. He received his bachelor's in biology degree from Elon University in North Carolina, his PhD at Florida International University in Miami. Before coming to Harbor Branch, he had visiting appointments at the Smithsonian Tropical Research Institute in Panama and the Perry Institute for Marine Science, which is based in the Bahamas. And Josh was also on the marine science faculty at Eckerd College in St. Petersburg before coming here in 2007. And when Josh came, he brought a lot of new skills and a lot of new talent, and you'll be seeing some of that tonight. He is currently an assistant research professor in Harbor Branch's Robertson Coral Reef Program, and he is a coral ecologist whose research focuses on coral health and the environmental factors that influence coral degradation in the wider Caribbean. And tonight you're going to see most of his work focused in Florida, but he also has worked extensively in the Bahamas and throughout the Caribbean. Many of you may remember his very first lecture last year in the series, which was entitled, "Who Slimed My Coral?" So let's welcome back Dr. Joshua Voss, who will speak on a decade of demise, charting the past and future of Florida's coral reefs. Josh.
Alrighty, first things first. Everyone hear me okay?
Yes.
Alrighty. This title sounds ominous. It's not as bad as it sounds, so that's why there's that question mark at the end. I was challenged earlier about midweek to make this as positive as possible. So we're going to go about 50-50. About half the talk will be doom and gloom, about half the talk will be new possibilities and positivity. But first, let's talk about what I'm not going to talk about. First and foremost, while I love pretty little fishies, and I love menacing sharks, and I love amazingly intelligent dolphins, this talk instead is going to focus on benthic ecology, on the organisms that make up the bottom, that drive the ecosystem, that enables these other charismatic megafauna to exist. So I'm going to focus on corals themselves, the animals, and their symbionts. I'm also not going to talk about politics or religion, because my mom told me that's not polite in certain company. And specifically, I'm going to avoid the question, do I, or do you, believe in global climate change? It's not an issue of belief. Faith is the realm of beliefs. Global climate change is a scientific fact, and so this is a question that I'm not going to address whatsoever in this talk. I'm also not going to talk about the cause of coral reef decline. As a coral reef biologist, I'm often asked, well, what's really doing it? What's that one thing that we could just fix that, everything would be okay? And the answer is, it's not just one thing. There's not a singular cause, so I'm never going to pin it down to just one thing. I'm going to present a couple different things this evening. And lastly, I'm most definitely not going to talk about throwing in the towel. You'll hear way too many people say, oh, Florida's coral reefs are shot. Let's completely change our focus and put our energy, time, effort elsewhere. That's not the case. Florida's coral reefs aren't lost. We shouldn't give up hope yet, and I hopefully will give you some indication of why this evening. Alrighty, so we have these amazing coral reefs here in the state of Florida. They extend about 220 miles from just south of us in Martin County at St. Lucie Reef near St. Lucie Inlet, all the way down and around the bend to 70 miles west of Key West into Dry Tortugas. Along that track, it's not a contiguous reef tract. That is, it's not a continuous ribbon of coral that runs the entire way. There are areas where there are no corals present. In addition, our reef is relatively young, about 5,000 to 7,000 years old. So most of the corals that we have here have only been in existence since the last little ice age, around 9,000 or 10,000 years ago. So geologically speaking, it's a fairly young reef. That said, for the spatial extent of the reef that we have and the spatial area of reef that we have, it's still fairly diverse, fairly productive, especially considering that we're at kind of the northern limit of where coral reefs can develop. Now, along this entire reef tract, 220 miles, nearly that entire area is protected in some way, shape, or form. There are a few areas that are not protected, principally those around Broward County, really have no protection to speak of. But most of them fall under jurisdiction of some kind of state park, local park, or the Florida Keys National Marine Sanctuary, or Biscayne National Park, or Dry Tortugas National Park. So there's varying levels of protection on different coral reefs depending on where we are along this big, huge reef tract. Some of those areas are absolutely no access. Research only, you can only go in there with a specific permit. Other of these areas, even areas that we would consider protected areas, are essentially not enforced whatsoever. So they're de facto unprotected areas. There's a range along this tract. Now, this is the one and only slide that I put up last year that I'm going to repeat this year. And that's because the importance of coral reef systems is something that needs to be emphasized when we're thinking about, okay, why does this all matter? So most of this is things you've seen before. I've added a bit more for this year. But principally, coral reefs are centers of high productivity. When I say high productivity, I mean two things. Number one, they're fixing carbon dioxide from the atmosphere. So a greenhouse gas, they're taking that carbon dioxide out of the atmosphere and putting it into a biologically useful form. Now that biologically useful form is then creating energy that can be passed to other different organisms. So effectively, these coral reefs are the base of a food chain. In addition, they're centers of very high biodiversity. And that's not just important because it's aesthetically pleasing. A lot of times we think of high biodiversity of, oh, that forest is so diverse, isn't it wonderful? Biodiversity has a couple of different key components that make it a biologically important function. First, when we think of biodiversity, we think of numbers of different species. The other thing we can think about is the kinds of different genes within those species. The reason this is important is the greater number of species that you have, and the greater the genetic diversity within those different species, the more likely that system is going to be able to handle change of any kind. If there's lots of different organisms out there, at least some of them are likely to survive in the midst of any large-scale changes. Next, they're important in coastal protection. This is particularly important when we're talking about hurricanes and storms here in the state of Florida. The tourism industry supported by coral reefs is estimated to be about $1.2, $1.4 billion annually in the Florida Keys alone. And related to that high centers of biodiversity, we have a lot of potential pharmaceuticals. Corals and most of the other benthic organisms are sessile. They're fixed on the ground. When you have numbers of organisms that are fixed on the ground, competing with one another for space, resources, nutrients, etc., they start fighting, to put it quite bluntly. And the way they fight is not the way we would think about it. They don't have appendages. They're not reaching out and trying to move each other. Instead, they're fighting chemically. So there's a lot of evolutionary biology going on where these individuals are producing compounds that are toxic to their neighbors. A lot of those compounds that are toxic to their neighbors could also be toxic to things like cancer cells. So we have all of these ecosystem services that coral reefs are imparting, and some of the more recent estimates of the value of coral reefs range from about 80,000 to 240,000 per square mile. So if we multiply that out quickly to the area of reef that we have here in the Florida Keys, or excuse me, in Florida, that means that our reefs are worth at least $20 billion. So we have this great resource, but there's been some problems. And so I'm going to outline some of the things that led up to this past decade. And then I'm going to focus on this past decade throughout the rest of the talk. But here, let's talk about how we got to this decade. One of the first big events in the history of Florida corals was a massive bleaching and mortality event way back in 1977, related not to heat, but to cold. Here is a quick article about the only recorded snowfall in Miami in 1977. And that same cold front that made it snow in Miami resulted in massive death throughout the Florida Keys Reef Tract and up along the southeast coast of Florida as well. We'll talk a little bit more about cold snaps when we get to the very end. In 1993, we know that there was a massive diadema, or sea urchin die-off, Caribbean-wide. It's believed that some kind of pathogen entered this system, quickly spread throughout the diadema population, resulting in mass die-offs on most reefs throughout the Caribbean, with estimated losses of about 98%. Earlier this evening, we were talking about how would something like this have happened today? Remember, 1983, there wasn't a Twitter, there wasn't a Facebook, there were no cell phones. And so when things started to spread, no one knew it was happening until almost after the fact. And that's the case with a number of different diseases. Throughout the 80s, we also had a number of epizootics. Epizootic is upon animals, whereas epidemic is upon humans. So think of an epizootic as an epidemic in animals. We had a number of different white band disease epizootics. And those of you that were here last week, you heard Dr. LaPointe refer to white band as well. White band is a disease that affects primarily two species of corals here in the Caribbean. Both of them are acroporids, or in the Acropora genus, elkhorn and staghorn coral. It's believed, and Rich Aronson up at FIT has done extensive work that proposes that white band was the major contributor to the acropora decline in the Caribbean. Then in 1995, we had a different kind of white disease that popped up, white plague. I've left a little peek here so you can see the difference between these two. Here we have healthy coral tissue on this acropora, a band of bleached tissue, and then bare coral skeleton. In white plague, conversely, rather than that band of bleached tissue, we go directly from bare coral skeleton to healthy coral tissue with very sharply demarcated sloughing line of tissue loss. Now, in addition to these gross physical differences, the thing that really made white plague troubling is that it wasn't just going after a couple species of coral. White plague was infecting nearly every species of coral that we find in the Caribbean. Subsequently, we've determined that white plague is probably caused by a number of different pathogens. It's not all one pathogen going after everything. Instead, a number of different pathogens can exhibit this same sign. White plague in 1995 knocked out about half of this coral, Dichostemia stokesi, or the elliptical star coral in the Florida Keys. And then in 1998, we have one of the most mass coral mortalities that has ever been recorded. we had an ENSO, or an El Nino Southern Oscillation, that brought extended hot temperatures to this area. And one of the interesting things about this event is that huge, massive, hundreds of year old colonies were dying off, in addition to small colonies as well. So this stack up, cold, disease, 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 hot, resulted in a situation by 1998 that things looked pretty bleak. We were to look at the living coral cover from 1996 to 1999. We're talking about, if we're looking at the bottom, the percentage of the bottom that is actually living coral cover. It's never been that high in the Keys, anywhere from about 15 to 20%, give or take. In 1996, it was recorded as an average of about 12%. And in just a short 3 years, we lost nearly 40% of that, dropping down to just below 8% total coral cover. So we see that we've experienced this dramatic decline. And in response to this decline, there were a number of different monitoring programs launched to try to understand what's going on. Which corals are we losing? Why is this happening? What could we potentially do to stop it? One of those programs was the Florida Keys National Marine Sanctuary Coral Disease, or Coral Health and Disease Cruise. It was launched in 1998. I'm going to focus on just the past decade. And it's an annual monitoring program that looks at 50 different sites throughout the Florida Keys, focusing on coral community structure, who's there, how big are those individuals, how are they connected in space relative to one another. And then secondarily, the diseases those corals are experiencing and any bleaching they may be experiencing. Let me clarify those a bit more. When we're talking about bleaching, we're talking about this breakdown in symbiosis within a coral. Remember that all corals are successful because they have endosymbiotic algae, little individual algal cells living within their tissues that allow them to gain light energy from the sun. During times of stress, and it's related to a number of different stressors, that symbiosis between the coral and the algae starts to break down. And there's now evidence that there's both chemical cues that the corals are getting stressed out and kicking the zooxanthellae out, as well as some very preliminary data from Dr. Sarah Edge here at Harbor Branch that potentially zooxanthellae try to get up and leave as well. Now, bleaching isn't always the end-all be-all. Many corals can recover from coral bleaching, especially if it's for a period of less than about a week. Two different things can happen. Number one, they can be recolonized by free algae out in the environment. Those algae can be ingested. Corals have molecular mechanisms so that they recognize those algae, and instead of digesting them, they put them in their tissues and use them as little photosynthetic organisms. Or, as you can see in this picture, often it's not pure white. There's still some areas where there's a little bit of those algae left. And so after the stressful event subsides, those algae can start to divide and repopulate that colony. So in other words, it can recover from bleaching if that bleaching event is short. If the bleaching event prolongs more than a week or so, more than about 5 days, effectively the coral starves. Remember, the coral's getting most of its energy from those photosynthetic algae. If they're not getting the sugar from the algae, they don't have a source of energy. So after about 5 to 7 days of continuous bleaching, corals starve and they die. In contrast to bleaching, disease is active tissue loss. So here you can see bare coral skeleton, live healthy tissue with the disease band advancing across, actively degrading that tissue in a necrotic fashion. Usually, bleaching doesn't persist that long, because if it does persist, the coral just dies. In contrast, a lot of these diseases can persist for years, very, very slowly killing that coral colony. Also, in contrast to coral bleaching, diseases have some aspect of transmissibility to them, particularly if they're related to a bacteria that's causing the disease or a fungus that's causing that disease. Those individual cells can move from coral to coral, passing an infection along the reef. This example, black band disease, and one that I'll talk about a couple other times throughout the talk, you can see a line of black tissue. It's a microbial mat that has a number of different organisms in it, mostly cyanobacteria and a couple of other sulfur bacteria as well. So we have bleaching and disease that we're looking at, and specifically we're looking at the spatial and temporal patterns of disease occurrence. Is it there or not, simply. Prevalence, that is the percent of the hosts infected. So just like we can have a prevalence of malaria amongst people, we can have a prevalence of a disease amongst corals. And we're looking at severity. The severity is a bit more of a tricky metric when it comes to corals. We can't look at things like white blood cell count. They don't have them. We can't look at things like corals describing their symptoms to us. How bad does it feel? They don't talk. So instead, we have to do things like amount of coral tissue that's been lost, or the rate at which that coral tissue is being lost. Those are good metrics of severity. In addition to looking at these patterns, we want to know not just what the patterns are, but why do those patterns exist? What's driving the patterns that we're seeing, and how might we be able to understand those patterns, slow them, stop them, reverse them, or at least understand why it's happening. And so we're going to look at some environmental factors, host factors, and pathogen factors that can be involved. Now, this first environmental factor, throughout the history of this monitoring program that's been done in the Florida Keys National Marine Sanctuary, concurrently there's been increased emphasis on looking at sea surface temperatures in this area. And it's not just maximum temperature that matters, but again, the duration of those stress events. So just like with bleaching, a coral can bleach and recover. With heating, if it heats up very quickly, stops, and comes back, corals can do just fine. But if you ramp up the temperature and then hold it at a very high temperature for a long period of time, those corals can't survive that extended stress. We can measure that extended stress through a metric called degree heating weeks. It's very simply a number that incorporates not just the extent of the temperature, but the time over which that temperature has been measured as well in a moving 12-week window. So if we look at this, this little moving chart up here, I'll let it run through. Come back around. This is an example of a degree heating week chart that starts back in September. You can see hot events shown here in orange and yellow down in the Florida Keys that we persist into late November and then December. You can see that it gets cooler and cooler. And we know that by January, we've had one of the coldest Januaries we've ever had. And we go right up until yesterday on the 15th, where there's no degree heating whatsoever. And this is a typical trend. If we were to simplify this into just looking at here, the year, 01 through 09, and the number of degree heating weeks, or the extent of prolonged high temperature during that year, we can simply graph this out. And you can see that 05 and 07 were relatively hot years. And this was true not just for the Florida Keys, but for the entire Caribbean. In fact, 2005 was the hottest year on record in the Caribbean, both in terms of absolute maximum and duration of high temperature. While all of these temperature measurements were going on, we've been out recording coral diseases through this survey, and we found that, again, coral disease can be variable. Here we're looking at the prevalence, or the percent of those corals infected that we know are susceptible to black band disease. And if we were to overlay those degree heating weeks with the years and the black band prevalence, you can very easily see that in 2005 and 2007, when we were experiencing relatively high temperatures for a prolonged period of time, that's when we see the highest prevalence of black band disease. So there's this fairly clear relationship between extended thermal stress, high thermal stress, and disease prevalence and coral mortality in Florida reefs. What's probably a little bit more disturbing is that Traditionally, corals got a break. During the winter time, as the temperatures would start to dip, much like they are now, black band disease infections would stop completely. When you went out to the reef, you wouldn't see any active black band disease killing any coral. After 2005 and that extreme stress event, we've been tracking it since that time, and while there's still a seasonal pattern, while there's still more black band in the summertime and generally less black band in the wintertime, a lot of these corals are never getting that break. So during the wintertime, black band is still persisting on some individuals. They're still losing coral tissue year-round. And we think that as this trends on for the next couple of years, it's going to result in more total colony mortality than we've seen before. Alrighty. So we've got this hot Earth. We've had this problem 05, 7. It's a continuing trend. And who knows what the Earth does when it gets too hot? It has hurricanes. It has its own built-in air conditioners. So when the Earth gets too hot, hurricanes come. So I'm going to talk about the hurricanes in 2004 and 2005. I recognize that some of these names will be four-letter words to those of you who have lived through them. So let's trudge on through. So in 2004, we had Hurricane Charlie, Hurricane Francis, and Jean, the ones that made direct hits here. I'm sorry, bringing up bad memories, I know. And Hurricane Ivan, which was a little nicer to us and just came back and hit us as a tropical wave. The interesting part about these is if you look fairly closely, none of them made direct hits on the Florida Keys Reef Tract. Hurricane Charlie skewed a little bit to the west. Francis was just far enough north to nail us, as was Jean. And Ivan, by the time it got down here to Florida, wasn't much of anything. So in 2004, we have this system where there's a lot of hurricanes, but not a lot of direct impact. And in fact, if we look at the corals themselves, there's very, very little direct impact of these hurricanes that's been documented on any of the reefs of the Florida Keys. Even the dry tortugas, which took a direct hit from Charlie, the eye passed right over Fort Jefferson. When they went out 2 weeks after and tried to document changes that had occurred on the reef as a result of Hurricane Charlie, there was no statistical difference. There was no significant coral loss from a direct hurricane hit. In 2005, things got even worse. We started off with Hurricane Dennis. Those of you that were here last week may remember that Dr. LaPointe talked about Hurricane Dennis being strong enough to right the Spiegel Grove from being on its side to all of a sudden sitting upright at 120-foot depth. So although this hurricane was strong enough to roll a ship 100 feet underwater, its impacts on coral were still relatively minimal. There was some minor bleaching, a little bit of sedimentation. Down in the lower Keys, there were some areas of acropora that were lost, but the overall impact was relatively minor. Hurricanes Katrina, Rita, and Wilma also came much closer to the Florida Keys Reef Tract than any of those hurricanes in 2004, but the story was still the same. The relative impacts from these hurricanes was fairly small, which is interesting. The question is, well, are these hurricanes then angels or demons? We know that hurricanes can cause tons of direct physical damage. The wave action can knock over coral colonies. It can fragment off branches. Sediment can get lifted up and deposited, completely smothering a reef. Sediment can blast through, ripping all of the tissue off of those coral colonies. But at the same time, remember, those hurricanes are nature's air conditioners. They're bringing colder temperatures to that area, reducing the water temperature. If we look here at Hurricane Katrina before and after in 2005, you can see the orange degree heating weeks in the Florida Keys during the last week of August. Immediately after Hurricane Katrina passed through at the beginning of September, you notice there's effectively no degree heating weeks in the Keys, nor is there any heating up this way following Katrina's path. So while these hurricanes bring the potential of physical damage, they also bring an amelioration or reducing of temperature stress. They're effectively lowering the thermal stress during this time period. And it's believed that Hurricane Katrina actually reduced the potential of bleaching in 2005 following its path. If we were to look at each of these hurricanes in 2005, you can see that after each one of them, Dennis, Katrina, Rita, and especially Wilma, right after each of those hurricanes, there was a pronounced drop in sea surface temperature. And particularly with Katrina, leading up to Katrina, things were looking horrible. Again, this was the hottest year on record for the rest of the Caribbean. We thought it was going to be one of these years where disease was running rampant, bleaching was all over the place, and we expected there to be a lot of coral mortality. But Katrina came through, it dropped all of the water temperatures, the degree heating weak stress fell, and the relative amount of bleaching and disease on the reef was fairly small. Now, this was a localized effect. In the rest of the Caribbean, in the Virgin Islands, for example, that heating stress continued. It was a Caribbean-wide event. Of the remaining acropora that were in the Virgin Islands, they lost about 50% during one bleaching event in 2005 alone. So effectively, Hurricane Katrina saved a bunch of our corals. So we've had all of these events going on. Remember that from 1996 to 1999, there was this pronounced drop in the living coral cover in Florida reefs. But if we look at 2000 through 2009, the drop's not that big. Here we're at about 8. Here by the end of 2009, we're about 6.5. So despite all of these hurricanes, despite this bleaching event in 2005, despite increased appreciation of coral disease, the amount of living coral cover that we're seeing has not fallen appreciably. So you'd think maybe at this point I'm going to switch to the happy part of the talk, but not yet. Because if we look at whether or not this is stabilized, if you ask the question of, has coral cover loss stabilized? Up until 2009, I would have told you, yeah, the amount of living coral tissue has remained relatively the same. But in just about the past month, we've had another cold front push through, not nearly as strong as 1997, it hasn't snowed here yet. But that cold water mass, and again, more importantly, the duration of that cold water mass in the Florida Keys has led to mortality, especially on nearshore reefs where the water is not as deep and temperature can change faster, at rates of up to 75% of the corals. Those of you that were here last week and listened to Dr. LaPointe's talk, someone asked a question about this cold water event. And some of the early estimates that were reported by Corey Walter down at MOV were anywhere from 20, maybe 25% of the corals have been lost. Her latest estimates, and again, this is kind of preliminary data, are that in those nearshore reefs, at least 3/4 of the living coral have been lost. In addition to continued losses, we still have this ongoing reduction in coral colony sizes. So we're having lots of corals that are experiencing partial mortality from a sedimentation event or from a disease event. And so they effectively become smaller. In addition, a lot of those large coral colonies, the old sentinels of the reef, are dying and being replaced by some of these smaller individuals. Now in corals, it's only when you get really, really big and really, really strong that you can reproduce. The small guys spend most of their energy just trying to make it. They allocate nearly all of their effort towards defense, competition, and feeding, or just getting space. It's not until corals get extremely large that they're released from that competition, and then they can put effort towards reproduction. So because we're losing a lot of these large coral colonies, the potential reproductive capacity of the reef is falling slowly but surely. And so we expect there to be continued reduced reproduction over the next few years. We also know that there's ongoing and new disease outbreaks. In 2008, we went out to the dry tortugas, and it looked like someone had taken a huge bowl of kind of loosely mixed plaster of Paris and just poured it over the top of huge coral colonies. Big, sloughing, dripping, white patches of tissue falling down the size of these colonies with completely bare skeleton left behind them. So there's still these new syndromes popping up that we don't quite know what they are. We also have a better record of persistent paling and bleaching. In the Florida Keys National Marine Sanctuary Survey, as well as other surveys conducted in the Florida Keys, originally people were looking at the numbers of corals there, whether or not they had a disease, whether or not they were bleached. We've literally started to appreciate shades of white better since then, so that many of these programs now include paling at different levels. Approximately 25% of the algae lost, approximately 50% of the algae lost. So the data from the more recent years indicates that while bleaching may not go on continuously, some level of paling usually does. And lastly, kind of the elephant in the room is a new problem with ocean acidification. We know that increased CO2 in the atmosphere is going to drive chemical changes in the ocean to increase the acidity of the ocean itself. And at this point, it's still very unclear how corals and other reef organisms are going to respond to this change in ocean acidification. So it's an area of concern as well. All right, but we do have hope. And in fact, I'm serious. We actually have New Hope in Florida. It's a little town up in the Panhandle. We may not want to claim it because it's the Panhandle, but it's there. And some of those areas of hope are, number one, we've seen diadema sea urchin recovery in other areas of the Caribbean. We haven't seen it here in Florida yet, but urchins have larvae that have fairly long dispersal rates. So they can move around areas fairly quickly. So the hope is that as these other areas of the Caribbean start to recover, some of the larvae from those could eventually end up in the Keys, and we might see some diadema recovery here in the Keys, but it hasn't been documented yet. Other hopeful area comes in terms of legislation. I know I was going to talk about politics, but this is just a little stretch. Back in 2008, we had legislation passed that effectively banned any new outfalls whatsoever in the state of Florida. In addition, there were two commitments made by 2025 that 60% of our wastewater would be reused in things like irrigation with reclaimed water. And by 2025, removal of all sewage outfalls in the state. Now, these are legislative measures. Does not necessarily mean these things will happen for sure. It'll depend, of course, on the political process between now and 2025. But there is hope. On March 31st, right at midnight between March 31st and April 1st of this last year, Delray Beach was the first municipality to literally turn off the faucet. They closed their sewage outflow and completely stopped dumping any sewage waste on the reef at their shoreline. So we're proud of them. Other things to be hopeful about, and again, Dr. LaPointe mentioned this briefly last week, acropora restoration. Both acroporid species here in the Caribbean, the staghorn and elkhorn corals, are listed on the endangered species list, primarily due to massive losses of these corals due to disease throughout the 80s. And actually, right now, there are 82 additional species under consideration to be considered endangered by NOAA. Now, NOAA is not the ones who make the final determination on what's endangered, but their recommendation carries a whole lot of weight. And because these two have been considered endangered already, there was a large amount of stimulus funding that has gone towards acropora restoration. About $3.3 million is direct stimulus money to this program, and about $700,000 or $800,000 is federal flow through the state to this program. And with this $4 million, what the Nature Conservancy, Coral Restoration Foundation, Florida Aquarium, and a number of other partners are proposing to do are expand the 4 nurseries in the state of Florida to 6 and increase the capacity and output of each of those such that within the next 2 to 3 years, at least 12,000 corals will be outplanted or put back onto the reef in a recovery effort. One of the keys about this, and one of the things that, that the Nature Conservancy has really brought to the table along with Florida Aquarium, is that the original coral restoration programs were like gardening. You go out and you put a whole bunch of things out there and you hope they grow and they look good and they look pretty. The problem is the long-term impacts of that are poorly understood. For example, we know now that farming salmon, you have to have a very important consideration of the health of the salmon you release and the genetic stock of the salmon you release. The same thing should be true for corals. And so in efforts to improve the quality of the corals that they put out, to increase their likelihood of surviving, as well as to make sure there's no kind of unexpected ecological disasters, they've adopted a program where they'll be doing both genetic and bacterial screening of any fragments that are outplanted. And again, Dr. Edge is involved in looking at the gene expression of some of those fragments to figure out what the optimal type of corals would be to outplant. Other things to be excited about. So we know that there's this problem with coral recruitment in terms of the number of new baby corals that are making it into the water column and then getting onto the reef. But corals are the kind of species that throw a whole bunch of eggs out there and hope that a couple of them survive. Maybe we can change the odds. If we had a better understanding of coral recruitment, that is, new corals getting onto a reef, we can potentially get them to not only settle, but survive to adulthood. So here's an example of corals spawning. They release their sperm and eggs. They float up in the water column to the surface. The sperm fertilizes the egg. And you have a new larvae that starts swimming around and eventually finds its way back down to the bottom. So here's a couple of individual little larvae. When they find a nice, happy place to settle, they quickly turn up on the end and literally metamorphose and flatten out into a new coral polyp. So here you can see the mouth and the tentacles around that individual little coral polyp that kind of looks like an anemone when it first starts out. Now, Rafford and Williams and Val is down here in front, Val Paul at Smithsonian Marine Station, have done a couple of projects where they've identified the settlement cues related to these coral larvae to try to identify what makes coral settle. That's useful for two reasons. Number one, if we know what's releasing those chemical cues, we can effectively engineer an area to increase the likelihood of coral sediment in that area. In addition, if we're doing any kind of restoration program where we're trying to get coral larvae from the environment, grow them up to adulthood, and then plant them back out after we know that they're going to be successful, you can increase your likelihood of gathering coral larvae using this technique. Alrighty, so we have a whole bunch of different corals and all these ideas to potentially outplant these corals. But the question is, which one of these are going to be the toughest? And not just who's the toughest, but why are they the toughest? And one of the interesting things is that even way up here at St. Lucie Reef, which is considered the northern extent of coral reef formation in the entire hemisphere, not only are corals persisting through hurricanes, persisting through Tropical Storm Faye, and all of the water that was released from St. Lucie Inlet during Tropical Storm Faye. They're not just surviving, they actually appear to be reproductive. They have gametes, sperm and eggs, developing within their tissues. We haven't been able to witness spawning yet, but that's the next step. We're partnering with Fish and Wildlife Commission to look at not just the health of these corals on this reef, but also the impacts of that freshwater discharge. And here, these two little guys, one saying, "I'm way more resilient than you'll ever be." This idea of resilience is a buzzword that you hear a lot when we're looking at different Florida reefs. And again, it gets back to that notion of biodiversity. Is there enough diversity within this system and enough genetic strength within this system that even after harsh times, those corals will be able to recover and persist? That's what we're trying to figure out. Another way to figure out which corals are tough can sometimes happen fortuitously. We were conducting an experiment down at the Moat Marine Lab on Summerlin Key with some funding from the Protect Our Reef Plate. And one of our interns, Dan Rowan, who was an NSF undergraduate research fellow, we were able to get a whole bunch of different corals from the Florida Keys National Marine Sanctuary Recovered Coral Nursery. So when the Navy decides it wants a new dock, instead of just coming over and knocking out the old dock and putting in the new one, they actually call the sanctuary and say, hey, we're getting ready to knock down this dock. Do you want the corals that are on it? The sanctuary sends out a team, and they physically take every single coral off of that piling or off of that wall so that they can be preserved, used for both restoration purposes and experimental purposes. So we were able to get a number of those fragments. We sliced all those fragments up so that we could do a number of different replicates, and then we decided to kill them all. We artificially exposed them to black band disease that we collected from the environment. If you can see here in this picture, we have a number of different fragments where we've dropped equal aliquots of black band disease on these fragments. And then we tracked the progression of the black band across that coral over time. We looked at bacterial changes, both in the disease itself and in the mucus. We looked at gene expression, and lastly, coral immune activity. All of this with the idea of trying to determine what the relative roles of temperature and light were in black band, because we knew that temperature was important based on some of those monitoring studies. What we found is that once you reach a certain temperature threshold, it doesn't matter how much hotter it gets. The black band just keeps on going along at the exact same rate. So rather than it being a linear relationship, where the hotter you get, the faster it gets, at some point it levels off. The hotter it gets, it doesn't matter. What we did find out, though, is that if you track these over time, you can't see these really well, so I'm just going to color them in for you. Over time, some of the corals progressed quite nicely. The disease would march right across those individual fragments. But on some of them, like this guy down here, even after 7 days, there was very little migration of that black band across the coral. And when we started looking and analyzing all of these fragments, we found that there were differences between the coral colonies that we looked at. Now, these colors down here are just the identifiers that we gave the corals. They're not relative to the coral color itself. We just arbitrarily called one of the colonies red, one of the colonies green, etc. But what we found was that some of these colonies, like the red and green, were very susceptible to black band infection. They couldn't fight it off. They died relatively quickly. Whereas other colonies, like the yellow and blue, and to a lesser extent this orange colony, they were able to resist the infection in some part. The infection still began, but it didn't persist that much at all. These corals were somehow able to fight it off. So the question is, how on earth does that happen? Are some of these colonies just generally more susceptible to black band? Are they predisposed to some higher rates of infection for some reason? We know that there's variation both among these colonies that we looked at and between different species of coral in the same area, but we don't know much about whether or not the genetic diversity that we see in coral species. For example, in this experiment, we were looking at one species in the genus Montastria. We know that the level of genetic variation within that group is relatively high. So one of the possibilities is that some of those corals had genes that made them strong and able to fight off infection, whereas other corals had genes that made them weak, less likely to fight off infection. That would be an example of some natural variability. Of course, the other possibility is prior exposure to stress, damage, disease, etc. And again, that one can go both ways. We can have acquired immunity to something like a cold virus. If you get that cold virus once, chances are you're not going to get infected by the same cold virus again in the future. But other diseases, say for example dengue fever, if you get it once, you are much, much more likely to get it a second time. If you get it a second time, you're dead. We don't know if in corals they get black band, and if they can fight it off, then they're great. Or if they get black band, they're more likely to get it a second, third, fourth time, etc. And so we're looking at both the gene expression and the immune activity, as well as some of the bacterial symbionts with Dr. Edge. Laura Midlar is a collaborator at University of Texas, Austin, and Dan Rowan, who I just realized is here down front. this evening. Alrighty. So, in addition to identifying all these potentially tough corals, another place we can look for corals that have better health status may be on reefs that we call mesophotic reefs. Mesophotic literally means middle light. So as we start to go down the front of a reef crest, down about 150 feet, 50 meters or so, the amount of light and the quality of light available starts to drop off. Nonetheless, a whole bunch of corals are growing there just fine. And in fact, the rates of disease in those areas are fairly low. We also know that there's not a whole lot of thermal stress as you go deep. There's less anthropogenic impact as well. So there's a potential that these areas are serving as refuges of coral larvae themselves. So that maybe, just maybe, the deep corals reproduce, they release their gametes, those larvae then eventually settle on a shallow reef, repopulating the losses that we've had on shallow reefs. So it'd be the same thing as all of our fish on this side of the room have died. We still have some fish over here. Let's see if we can get a few of them to swim across to this side of the room. Same idea, but we're moving from a deep to a shallow reef, respectively. Part of this program has been funded by the NOAA Cooperative Institute. Those of you that saw Dr. Pomponi's talk earlier this semester heard a bit about this. And one of the projects that we're pursuing is looking at these mesophotic reefs, determining, number one, where are they and what do they look like? Trying to find new areas of mesophotic reefs and characterize what kind of corals are in those different areas. And then secondly, we want to know, is there any connectivity between these shallow and mesophotic reefs? In other words, is there genetics that are moving from one area to another? Are there larvae moving from one area to another? We don't know yet. And then lastly, if these areas are going to be something for us to worry about, if they are important not just for this idea of refugia, but also for the ecosystem services that they have, we need to know what their health status is. There's very little work done on these mesophotic reefs to determine, do they get the same diseases as shallow reefs? Are they susceptible to bleaching, etc.? And because mesophotic reefs seem to be in better condition right now than shallow reefs, it makes sense to be more proactive about identifying them and protecting them than we have with shallow reefs. Alrighty. So, all of these things are going on that we can be positive about, and these are all things that various different researchers are doing. Nonetheless, corals still need a lot of help. There's still a lot of things that are leading towards coral degradation. And so I want to end with a couple things that you personally can do. Number one, reduce the pace of climate change. Climate change happens. It's an inevitable fact of geologic history on Earth. What makes this climate change unique is that it's happening at a rate that's unprecedented, and it's at a rate that none of the organisms on Earth can evolutionarily cope with. So if you're interested in how you can potentially reduce the rate of climate change, check out carbonfootprint.com. It's a way to get a handle on how you yourself are contributing to this problem, or how you yourself are helping to ameliorate this problem. Reduce nutrient impacts. We know for sure that nutrients can, number one, exacerbate coral disease. Number two, if a coral becomes injured, if excess nutrients are around, it takes it longer to heal. And number three, excess nutrients can contribute to overgrowth by algae. So, simple steps. Who lives within a mile of the beach here along the coast? Simplest thing you can do, stop fertilizing your yard. We love grass. We love all these amazing pretty plants, but doing a simple thing like converting towards native vegetation and reducing the amount of nitrogen and phosphorus that you put on your lawn can reduce the amount of nutrient inputs in nearshore waters. Likewise, and I'm just as guilty of this as everyone else, I'll admit it, but those of us that have pets, collect those little presents that they leave for you and compost them. Alrighty, responsible boating and diving. This is something that has received a lot of increased attention here in the state of Florida, but it's still an issue. We still have over 50 to 60 boat groundings a year. Most of them are not due to negligence, they're just due to basically people not knowing where they are. I guess that is a form of negligence, but it's not intentional. People just aren't paying attention. So improved education on using charts, using a GPS, and likewise improved education on diving and not standing up on a coral, not touching a coral, etc., are things that we can actively do. Well-engineered coastal development. I won't say stopping coastal development because that would be a little too optimistic and a little hypocritical considering my wife and I just bought a house that's right by the beach. But a lot of the coastal development that goes on can be engineered better. Rather than having seawalls, keep the dunes. Move the houses another 100 feet back from that area. When we have an area where beach renourishment is suggested, think about whether or not that's a long-term solution or simply a short-term way to keep sand on the beach for this one more season. Support marine protected areas. This is something that has gained a lot of traction, especially in the last 5 to 10 years. Initially, marine protected areas met with a lot of debate, especially among those who are subsistence fishermen, those who rely on fishing for their livelihood. There's a number of different marine protected area models. When I say support marine protected areas, that means you also have to be willing to support the enforcement of those areas. Simply having them isn't good enough. We actually have to enforce the legislation that's enacted to create them. Avoid eating grouper. Grouper is one of the most overfished groups of fish. It's tasty, I love it, I'll admit. But whenever you're given the option, go for the mahi instead of the grouper. Groupers are extremely slow to reproduce. They take a long time to get from juveniles to adults, and their overall life history is relatively slow. So when we overfish them, it takes them a very long time to recover. Mahi, on the other hand, mature very quickly. They reproduce in higher numbers, and so they're less susceptible to the overfishing pressures that we put on them. So when you order that sandwich, ask the waiter if you can have mahi instead of whatever else they give you. And lastly, learn more and share your knowledge. It's very, very simple to come to a forum like this and sit back and go, oh man, that was great, or not so great. It's much more difficult to then go out with someone else that either you know or someone else that maybe you don't know and help them to understand why you're concerned about these issues as well. And if you want some more information and facts that you can share with them, be sure to check out Harbor Branch's website. And with that, I'd like to thank a whole bunch of different people that helped me with the work. The funding sources, primarily the Banbury Fund that supports the Robertson Coral Reef Program here at Harbor Branch. And if you have any questions, I will be happy to take them.
Can you get a flu shot for the coral?
Can corals be immunized? I got the first part of your question.